Now we are taking up the process of the fertilization. Now if we take, this is the ovary, so this is the branched ovary in which the ova are formed and they are coming down into the oviduct. So they are coming from the oviduct downwards. Now what happens? The sperms can reach these ova in two ways. So the first way was that if it is self-fertilization, I am taking a green color for this. If it is self-fertilization, they will be coming from the uterus down and they will be coming towards the ova by chemotaxis. So ultimately they will be reaching the oviduct. So this is the case if there is self-fertilization. But if it is cross-fertilization, I am taking red color for this. If it is cross-fertilization, then the sperms, they have entered the Lawler's canal from here. So that means the sperms of the other Fasciola hepatica, they have entered the Lawler's canal. So they will be coming down into the Lawler's canal and they will be reaching the eggs, just like this. So ultimately, either the fertilization is self or it is cross, the sperms will be reaching the oviduct. Now this is the part where the fertilization will occur. So this is the part of the oviduct, or we can say the fertilization occurs in the terminal part of the oviduct, at the very end of the oviduct. So here what happens, that a zygote is formed. So I am taking a cream color for this. So students, this is the zygote formed in the very center. So the sperms, they have fertilized the eggs and a zygote has been formed. Now if we draw a zygote, suppose like this is the zygote formed in the oviduct. And from the lower side, I am taking a blue color for this. Suppose this is the median vitelline duct, and what is coming upwards, in the very center we have the yolk reservoir. So this is the yolk reservoir, and from here the yolk is coming upwards and it is reaching the zygote. So when it reaches the zygote, yolk is poured over the zygote. So this is the layer of the yolk, the blue layer. So in between we have got the zygote. Now what happens? Now the secretion of the Mehal's gland, it is performing its function. So its function was to harden the capsule. So what happens when the secretion of the Mehal's gland, I am taking it in black color, when it is pouring over this particular capsule, this capsule becomes hard. And the secretion of the Mehal's gland, it is also serving to lubricate the path of this capsule. Now it is not called as zygote because we have the zygote in the very center. So we have got the zygote in the center. We have got a yolk membrane over it, and this is now called as a capsule. Now these capsules, they are now going towards the uterus. They enter into the uterus. Just see, this is the black line. They are coming towards the broader part of the uterus, and now here in the uterus they are stored now as capsules. So that's why we say that the uterus is full of capsules. So this is the process, this is the physiology of the fertilization and the formation of the capsules inside the Fasciola hepatica. Now we have to consider this capsule into account whenever we have to take the further development of the fasciola hepatica. Now students, we know that the capsules, they have been formed inside the uterus of fasciola hepatica. Now here you can see the number of capsules which are present in the uterus. So this broad part of the uterus is also called as the metraterm, and this part is muscular, and when this muscular part of the uterus it contracts, these capsules, they are thrown out from this uterus part, and they are coming out from the female genital tract. So ultimately what happens, that these capsules, they come out from the female genital pore. So this is for the dissemination of these capsules. Now what happens further we'll be discussing in the next part of this video. So I hope that the process of fertilization, the cross and the self cases, you have understood those cases, and what is the physiology of the capsule formation, I think you have understood too. So in the next part, we are taking up the further formation of, or you can say the life cycle of fasciola hepatica. Now this diagram, it is showing the further development inside a capsule. Now students, the slide A or the figure A, it is showing a single capsule which is present in the uterus. And these types of capsules, they can be like 3000 in number. So there can be like thousands of capsules stored in the uterus of one fasciola hepatica. And in different cases, it has been seen that near to 200 flukes can be present in a single sheep. So as this fluke was present in the sheep, each fluke can have near to like 2000 or 3000 capsules, and one sheep can have like 200 flukes. So that means they are in lakhs. So the amount of these capsules, it is very prevalent. It's very huge in case of fasciola hepatica. Now if we see the structure of this particular capsule, then we can see that first of all we have this operculum. So this is a very important part of the capsule. So here you can very well see the operculum is the cap-like structure which is present over this capsule, and this operculum can open out. So I'll be taking it later on. So then we have a zygote which is present in the very center. So this is the zygote which is present in the very center of this capsule, and it is surrounded by many yolk cells, and this is the egg shell. So this is the shell of the capsule which was hardened by the Mealy's gland secretion, and this shell is formed again, it is formed by the yolk itself, by the vitelline glands. So students, what happens next is that this particular zygote in the very center, as you can see in the diagram B now, it divides into two cells. The lower cell is called as the somatic cell, and this is larger in size as compared to a upper propagatory cell. This is a smaller cell which is formed by the division. Now these two cells, they are present inside the capsule, and this capsule is present inside the uterus. Now the upper propagatory cell, it is forming the whole larval body. Now this is very important to note. The propagatory cell, it is forming the larval body, whereas the second cell or the somatic cell, it forms the germ cell mass. So this is a very important thing to note. So here you can very well see that you have the yolk cells, you have the operculum, and this is the embryo which is formed by the propagatory cell. Now this embryo, it develops into the very first form which is called as the miracidium. So students, this is the first larval form which is formed inside the capsule. You can very well see a single miracidium, and this miracidium is now present inside the capsule, and it will be coming out whenever the operculum opens. Now how the process goes on and how the development occurs, how the life cycle occurs, I'll be taking up in the next part of the video.